Hi, everyone. Thank you for joining us today for this webinar that we're holding with CATS Foundation, Niemann-Pick UK, and we're really pleased to be joined by Azafaros. So what we've decided to do over the course of the next few months is hold a few regular webinars of our different communities to talk about some really important topics that have come up and we're always being asked questions about. So as you'll know from today, we're here to talk about placebos, placebo arms, and why they're going to be used in clinical trials. So before we get started, I thought it'd be very important for us just to know who's here and who's going to be speaking. So I'm very pleased to say that Chris, who's the CMO from Azafaros, is joining us. Will, who's a trustee and ex-chair of Niemann-Pick UK. Toni, who's the CEO of Niemann-Pick UK, and myself, who's also the CEO of the CATS Foundation. And all of us have a real connection to the our different rare disease communities. Again, I think it's important for everyone to know why we're doing this as a joint webinar. So CATS and Niemann-Pick UK are both members of an organisation called the UK LSD Collaborative. And what this is a group of rare disease advocacy groups in the UK who are all supporting families affected by lysosomal storage disorders. We really believe that working together and working collaboratively can not just benefit our own communities, but also the wider LSD community and help drive forward those treatments that we're all trying to really find for our different families we support. Again, one of the real key things we're doing as groups, individually and collectively, is supporting and empowering our families. We really believe they need to be given enough information at the right time to help them make the right decisions when it comes to terms of looking after family members, making decisions about schooling, making decisions about clinical trials they could potentially take part in. And we do that by providing that information and educating them. And we believe that's such an important step that we are really committed to doing, to making sure that they have access to the right information at the right time. As I mentioned before, collaboration is something we're very, very hot on, not just at CATS, but Niemann-Pick UK, the LSD Collaborative generally, and also Azafaros. And we're really pleased to say that working together can help drive forward not just that area of support, but the area of clinical studies and clinical trials. And the voice of the patient has never been more important, especially from the regulatory side. And that's something that we've worked tirelessly on to do to make sure that our patients, our families, and everyone is heard when we come to look at developing clinical studies for our different communities. So I mentioned before, these are the speakers who will be joining us today. And we have a really exciting agenda, actually, just to talk through the topic of placebos. And what we've decided to do is actually break this down into a little bit more detail. So Will and I will be talking through about what actually is a placebo. I think this is a topic that comes up quite regularly, but not many people fully understand the history to why they're used in clinical studies. Then we touch on why placebos are used in clinical studies. And this is where Chris is going to step in and he's going to talk through from the Azafaros perspective and also industry in general. And finally, we're going to be looking at what is actually the impact on the patient and the family. And this will be a really interesting discussion between Toni and Will about actually making that decision to participate in a study with a placebo arm, but actually are people given the right information to make that informed decision about whether they should be joining that particular study. So I'm really pleased to say we decided to put a goal today to make sure that everyone knows why they're joining the webinar, and when they step away, they, in theory, we hope would have learned something. So our goal today as a group is to make sure that everyone understands why a placebo arm is used in a rare disease clinical trial, why they're used, and the choices that families have to make. And I think that's going to be a really important topic for us that we're going to touch on over the next kind of 55 minutes of this webinar. So on that note, I'm really pleased. I'm going to introduce Will, who's going to join me on the screen. So hi, Will.
Hi, Dan.
Thank you for joining. So I thought first up, it would be quite good for everyone to have a little bit of an introduction to you, for those who don't know who you are. And then we'll fire on some questions about placebos. It'd be quite cool to give an intro.
Sure. Thanks, Dan, and thanks for the opportunity to join this webinar today. So, yeah, my name is Will Evans. I'm the father of Sam, who is 15, and he has Niemann-Pick type C. And I've had involvement with the charity since he was very small. So I've been the chairman of Niemann-Pick UK. I've recently stepped down from that role, but I remain a trustee on the board. And I've been involved with Niemann-Pick UK and their scientific subcommittee and research subcommittee, and been quite heavily involved with drug development and clinical trials in Niemann-Pick type C. outside of those roles with the charity, my day job is I'm a GP, but I also work 1 day a week in seeing patients in a clinical genetics department in Yorkshire, and I also do some research at the University of Nottingham. So I wear several hats.
Awesome, thanks, Will. So it's good actually that you can join, because you have this unique position of not just being the parent of the family, but seeing it from the medical side. So you can see the challenges that some of us as families have experienced over the years, but also from the medical side, which I think is unique, and we're really pleased that you could join us today. I think the first kind of question, it'd be quite good for you to, if you can, just to give us a brief overview of actually what a placebo is, because this is something that many of us are told about when we go to clinics. We talk about clinical studies with our doctors, and I don't think many of our families really appreciate and understand what a placebo actually is.
Sure. So a placebo is essentially something that is given normally in the context of a clinical trial, so that one can compare the effect of a drug upon somebody, as opposed to not giving the drug, but trying to make things as fair and as balanced as possible. So I'm trying to remove a thing called bias. So I think in some respects, it's almost easier to go right back to the start. And a very simple question is, how do you know that something works or doesn't work? And there's a few things that we can, when you're trying to approach that. Sorry, I think my fire alarm's going off, but there's someone else in the house, so
That's right.
Hopefully I'll get a knock if there's a problem. So if you start off by trying to work out, well, in life, how do you know something works? And the best way to do that, and certainly in the point of some form of health intervention, is to essentially get a group of people who are as similar as they can be, and then to try and separate those people and into two groups, and give half the group something that you think is going to have some beneficial effect, and not give that to the other half. And then you can compare the outcome between the two. And to a certain extent, people have been doing this for several hundred years. There's a guy called James Lind, and he was a ship's doctor, and he took a load of patients who were on his sailors who developed scurvy, because that was vitamin C deficiency, really common in the 18th century on ships when they were out at sea for a long time. And he took people who were as similar as they could be, both with the same degree of scurvy, and then he gave some of them a mix of essentially vitamin C and vinegar and a few other things. Half the patients that, and half he didn't give that to, and saw the outcome. And the outcome in that was that the vitamin C and the vinegar improved things. So that's really where it comes from, that idea that you want to basically see if something works and compare it with two as similar groups as possible. So the first thing you do, if you've got a large group of people, you want to divide them as similarly as you can, and you want to do that randomly. You don't want to have any bias by you cherry picking, going, well, I think he'll do better with the drug than that person, so we'll put him in and not that person, because then that's not a fair test between the two things. So you want it to be not biased. You're not cherry picking the people, and you want the people to be as similar as possible. But then everything that you do to those two groups of people, you want that to be absolutely as similar as possible, other than the one thing in question. So if this is a test to improve people, a special drug that improves people's walking, but the group that you give the drug that improves walking to, you also give them physio 5 times a week, and the other group go no physio, that's not a fair test. So everything that happens to the arm, to one half of your trial, should be given to the other. Now, one of the key things is, well, hang on, they'll know if they're receiving the drug, because the groups aren't quite the same. They're being given a tablet, or they're being given something, some form of intervention, which this group isn't. And that can cause bias. So it can cause bias to the people receiving the drug, because they know they're getting something that's different. And there's lots of evidence that even when you just give people a sugar pill or an injection of water or a sham operation, their symptoms can get better. So, and that's known as the placebo effect. So you want those people you want the other group who are not getting the drug to be having, in every other way, the same experience. And essentially that's what a placebo is. A placebo is giving something so that the experience of the two halves of the trial is exactly the same for the patients, but also even for the doctors giving it. So if the drug in question is a pink pill that's square that you take 3 times a day, then the placebo people will get a sugar pill or an empty pill that looks exactly the same and is given in exactly the same way. And if this is an operation, and these trials have been done, they did one for knee arthroscopies. The patients who weren't having the arthroscopy went into a theatre, were put to sleep, and someone made 2 little nicks on their skin and put a couple of stitches in afterwards, so that when they woke up, they'd had exactly the same experience as the people who'd actually had the operation on their knee.
That's interesting. I've never heard of it from the perspective of the operation side, actually. And surely thinking about that in terms of the rare disease space, one of the challenges we have are patient numbers, as you know. So a lot of these studies do struggle when it comes to recruiting patients. Is that a common problem in terms of developing studies when it comes to finding patients in rare disease space when they're then having a placebo arm in their clinical trial?
Yeah, so I think there's a real tension, I guess, and this is how I see it in the rare disease space. So one is, you're absolutely right, you've got very few patients, so you want the trial to be as attractive as possible. And if you're going to enroll in a trial where you're told, and often it's not 50-50, it's not a divide quite as simple as I said, it can be 2 to 1, so 2/3 of patients are on drug and 1/3 aren't. But if you're going to say you're going to invest all this time, you're going to come regularly to the hospital, you're going to go through all this, and actually you've got a 1 in 3 chance of not actually getting the drug, that makes it seem quite a lot less attractive to someone involved. So that's in a way a kind of like a, something that is something that it makes you want to be involved with the trial less. But equally, as part of the nature of rare diseases, it's actually quite hard because the numbers are quite small to really make the 2 groups look as similar. So certainly in the case of Niemann-Pick type C, it's a hugely variable disease with not very many people. So if you randomly allocate people between the groups, just by chance, if there aren't very many people, some people with milder disease can be on one side than the other. So the putting a placebo in tries to make that as robust as possible. In that, because it's very hard to make 2 groups as equal as possible, at least you're trying to minimize that bias that can come from people knowing that they're on drug or not knowing or they're not on drug.
So that's really interesting, because sometimes I find that within our own communities, we get so laser focused on what impacts us in terms of our community, our struggles and our challenges. We don't actually realize a lot of what we're experiencing, such as recruitment for studies at placebo arms, or potentially those difficulties are transferable across many other conditions and many other diseases. Why I'm glad, obviously, we're doing the webinar together with the Niemann-Pick UK and the CATS Foundation, so we can really share that the challenges that we're facing, not just on placebos, but actually on how information is shared and how we educate and empower our community so they can make those right decisions. I think a really important, an interesting topic to discuss would be the whole process then about placebos, the history to them, especially in the rare disease space. Have you seen it from a long time ago? Because obviously you've been working within the medical profession for a few years. Is it something you've always seen happen, or is it a relatively new thing?
Yeah, so in the sort of full history of medicine, I guess it's a relatively new thing, but they have been around for quite a long time. So in a way, the kind of gold standard evidence is always what they call a double-blinded randomized controlled trial. So what we mean by double-blinded, and this is a key bit, is that neither the patient nor the doctor or the person doing the assessments knows whether they're on their drug or they're not on the drug. And that's a gold standard. And that is absolutely, if you're talking about blood pressure tablets or diabetes tablets or certain cancer treatments, that's absolutely the gold standard that one should be always expecting, because you've got lots of patients. So you can nicely and randomly allocate people between, and in actual fact, most the groups will be suitably similar just by the sheer numbers. The challenge we have in our rare disease space, as I've already said, is the numbers are small. So not only does it mean it's challenging to recruit, it's actually challenging to get a true fair test between equal groups. So trying to minimize and make the test as fair as possible on other things, such as blinding, is a really important thing. So I think that's really key, is if you're going to introduce a placebo, it's really important that not only the patient is blind to what drug, whether they're on drug or not, but also the clinician, their doctor, if possible, and also the person doing the assessments is also blind. Otherwise, we know, and this has been shown before, that if in a trial, there was a trial of multiple sclerosis drugs or a neurological condition, and the clinicians two different clinicians did the assessments, and the clinicians who knew that they were on drug or placebo scored the people who'd received the drug better than those who'd been on placebo, whereas the blinded assessor actually found that there wasn't any difference. So even when you're trying not to, it's really hard to fight our own intrinsic biases.
That's super interesting. Actually, I'm sure that I didn't know that, and many people out there won't have realised that that scoring side where the person administering the drug doesn't know as well who's on drug and who's not on drug. And I think that this understanding that it really is random, there is no predetermined who's on, who's not on the drug. And I think that's sometimes something that people struggle to understand when it comes to the placebo studies.
Yeah, no, absolutely, and I think that blinding is really key. However, and maintaining that blind as best as you can is an important thing. So making the drugs look the same, making them taste the same, making them so that if you're on, if people are going on and communicating, they say, oh yeah, mine tastes of strawberries and yours tastes of strawberries. I think the challenge can sometimes be that for certain things, it's impossible to double blind. So for example, we talked about that operation. Obviously, the person doing the operation knew whether they did the operation or just put a few nicks in the skin. So in that case, the doctor couldn't be blinded, but the patient was. In the case of the trial that my son was on, which I think we're going to talk about a bit more later on, it was a drug, but it was delivered via a lumbar puncture. So we were blinded to whether he actually was receiving a lumbar puncture or not. They basically did a sham procedure for it. Didn't actually do the full lumbar puncture, but they went through the process right up until that point. But obviously, lots of people knew that they weren't doing a lumbar puncture. And you can imagine working in a busy hospital, making sure everyone knows who's on drug and who isn't is really difficult. And sometimes drugs have side effects as well. So, if you know that everyone taking this drug the day after gets diarrhea and you're not getting diarrhea, you can quite quickly see how that blinding, the blinding of the placebo disappears because you know that you're not receiving the drug.
It's a much more complex topic than I think we all think at the beginning. We start digging into the details and actually how many factors are involved in understanding the whole process about developing the study with the placebo arm and then administering the drugs. That was a very interesting overview of that. We'll really appreciate that. Just on timing's sake, we're going to bring Chris in a second, who's then going to talk through placebos within an industry perspective, and you're going to be joining us again later with Toni to talk through your own personal experiences. So thank you very much for. If there's any questions, can people please just put them into the chat, and we'll pass them through to the speakers. So I'm now going to bring Chris into the conversation. So, hi Chris, how are you doing? You just need to unmute yourself very quickly. Chris, you just need to unmute yourself.
So sorry about
Perfect.
that. Yes,
That's right.
Thank you very much, Dan, for inviting me and allowing me to speak here, enabling that. Yes, I'm Chris Freitag. I'm Chief Medical Officer, which is just a very pompous way of saying I'm the physician looking after the clinical trials at Azafaros. And I'm very glad to be talking about this subject, which can be quite controversial. And as the person responsible for designing studies, it's really quite close to my heart. So I will hope over the next few minutes give you a little bit of a company perspective. Is that all right if we kick off, Dan?
Yeah, of course. I'll leave it to you. You just tell me next slide and we'll flick on through.
Great. I think we can go to the next one. Thank you. So this is going back a little bit, but I hope building on what Will has been talking about. So generally, how do we do clinical development? I won't make this too long, but just to give a few concepts. So the first part of developing a medicine is to do preclinical studies. That's in testing in cells, and not everyone likes to hear it, but we also do test in animals, of course, because we are obliged to. Then we go into phase 1, which is a study in which healthy volunteers give us their time and sometimes their patience as well, because these studies can take a little while, to try a drug and to test the basics of tolerability of it. But this is healthy volunteers. This is not in patients, and this can't be done with every drug. For instance, in cancer treatments, you can't give that to healthy volunteers, so it's generally avoided there. But phase 1 can be done in healthy volunteers in many instances. Phase 2 is the first introduction into patients, and here we do something called dose finding. So we generally have a rough idea of which dose is reasonably well tolerated from phase 1. But in phase 2, patients are different to healthy volunteers, as I'm sure you all know. So we have to just check again to our tolerability issues, and also sometimes in phase 2 we can start detecting some signs of effectiveness of the drug, what we call efficacy. Then phase 3, that's really the where we say the rubber hits the road. This is where it gets very serious. This is where we are doing the study in order to get approval from governments, from government agencies to put the drug on the market. And these studies are very comprehensive. They look at both efficacy, so whether the drug works, as well as safety, how well it is tolerated. And then phase 4 is really what we call post-marketing, after the market authorization. Sometimes it is advisable to follow patients for longer, even when a drug is approved on the market. And also, this can be a useful area in which we can do some cost calculations, how much costs are being saved by the treatment, and of course, continuing with drug safety. Moving swiftly on to the next slide, please. That's just an overview of how we do these studies. And the introduction of a placebo only makes sense in certain instances. So what we really need to do is we need to establish the safety and tolerability of the medicine, find the optimal dose, and show efficacy. That's the whole idea. Now, we can think up wonderful ways of doing this, but in general, we have to really agree this with different parties, if you like. And on the right-hand side, we have the who is involved part of the slide. So universities and labs sometimes give us the original compound, or sell us generally the original compound. Then we, as the lab or pharma company, take that further. And in the interaction with the treating physicians, with you, the patients, via the patient organizations, with regulators and payers. So regulators are the regulatory agencies that the governments put in place to protect the public and to ensure that only the right medicines get approved. And the payers are those people in charge of the purse strings. You may have heard of NICE, the National Institute of Clinical Excellence. That's the governing body in the UK that looks after these the pricing of drugs, to make sure that the pricing doesn't go too crazy. So when we design a clinical trial, we have to take all of these things into account. It's not just our thinking, well, this would make sense and this is logical, and why don't we take this approach? We have to really agree, and particularly with the regulators, with the government agencies. I'll come to the next slide, please, and then I can. So this is the dose finding, and this is, I won't repeat what was said before. What is a placebo? In this example, in the study, there are 3 different doses: 10 milligrams of a drug, 5 milligrams of a drug, and no drug X at all. But the tablets, as you can see, all look the same, and that's what the placebo does. So it's a medication that doesn't contain any active substance. If we go to the next slide. this is, I won't spend much time on it because we've really done, and Will did an excellent job in talking about what a placebo is. And also injection, tablet, capsule, even sham procedure, as well as said. What I just want to spend a little bit of time on is the placebo effects. And I've put it here on this slide as the sum of all effects in the placebo group. It's a bit vague. You kind of ask yourself, what does that actually mean? So the one thing is the act of taking a tablet can already induce a sense of feeling better. At least something is being done, and then the psychological effect is doing better. I'd also like to introduce a slightly different concept, and that is the fact that participation in a study already in itself can induce an effect. Now, I'm thinking we're speaking mainly with people who live in the UK and are familiar with the National Health Service, the NHS. But if we go to other countries like the US, where medical care is quite expensive, needs to be paid for, and insurances are not quite as widely covering as the NHS is, it makes a very big difference to a patient whether they get an annual visit at cost with their treating physician, or they get regular monthly visits in the course of a clinical trial. So the amount of exchange with a physician, not just physicians, maybe sometimes even more importantly, the physiotherapists and the speech therapists, occupational therapists who engage in these clinical trials can also, in and of themselves, induce a benefit. So if you have no direct comparison, you don't make the study exactly the same for all participants, whether they're receiving treatment or not, then you could introduce this bias, this effect of participation in the study, as well as the drug effect that Will has already described. If we go to the next slide, please. I'm just conscious of time. I don't want to take up too much time. So why do we do this? We want to avoid this bias, this undue influence that I've mentioned just now. So we need to generate this so-called high-quality data that supports regulatory approval. These are, again, the regulators, the government agencies. In some cases, it allows us to identify the natural cause of disease without treatment. And we can show statistics. We can use numbers to really demonstrate very clearly in graphs that there is an improvement. There's a separation of the curves, if you like. So the placebo is still seen as gold standard and is often requested by regulators for the reasons that I've just said. So if we, for instance, take the scenario where we would like to run a trial with treating the drug and using to compare to that what we know about the natural history of the drug. There, one could possibly argue, well, your patients in your study, they got the drug, but they also got other additional perceived or real benefits, which is the regular contact with the hospital, etc. And that's why they feel much better than your control group, and it has little to do with the actual drug. And to avoid this kind of interpretation, some would say misinterpretation, but whatever it is of the results, it is important to be as neutral and have really, really equal patient groups to treat and to apply the analysis to in these trials. We go to the next one. So I've belabored this a little bit now, but we can identify the natural course or course without the drug. We can get clear response versus, and in response to the treatment, and with a direct comparison. And the same also applies for safety, by the way. So if we see quite a few headaches, or in this case, if we're talking about Niemann-Pick C or neurological illnesses, seizures, for instance, is quite an important one. If we see an increase in seizures in a population, is that the natural course of the history that patients are getting worse? Is it part of the underlying illness, or is it the drug? If you can compare that directly to patients that were followed in parallel over the same amount of time, you can make a pretty clear statement on what this increase in seizures is likely to be due to. Although I have to say, and I thank Will for pointing that out as well, in rare disease, it's quite difficult because we rarely get very high patient numbers in our studies. And I think that's pretty much it. I talk a little bit about the challenges here. we don't like, and a lot of we don't like offering to patients, and patients don't like being offered inactive substances, especially if these diseases are very debilitating and irreversible. So you have the feeling you're losing time, and you need a certain sample size also when you're doing placebo comparisons. So you need to have a certain number of patients in the study, but I'll talk more about that later. In brackets on the right-hand side, the first bullet point says experimental drugs do not all yield positive results. I do want to just point this out. It's quite regular that studies are conducted and they quote unquote fail. That means that patients, I try to say to patients to not be maybe too upset when considering entering a placebo-controlled trial, because while it may be, placebo-controlled trials are generally done with products that are not proven yet. So it's not as if the patient is missing out on a proven treatment. Admittedly, missing out on the opportunity of getting a substance that could potentially be active, but at the beginning of the trial, that is certainly not, it's not a given. So that is something to also consider when thinking about placebo-controlled trials. If we go to the next one, please. So these are the considerations that we have to think about when we are designing the studies, and this falls on me. That's why I'm pretty close to this. But to think, how long does it have to take? How many patients do you include? And this is a feature of how rapidly the disease progresses and how consistent the disease is. And I think both Will and Dan have already alluded to the fact that with a lot of neurodegenerative diseases, including Niemann-Pick C, including the gangliosidosis, the clinical picture can be quite varied. And also not what we call a linear progression, so not a very predictable progression of the disease. The disease may worsen and then stay stable for a while and then worsen again. And these factors, the lack of predictability and the inconsistent course of the disease, can mean that the study needs to take a lot longer than in a very predictable disease. In theory, to the next point, how many patients need to get placebo in your study? In theory, one can decrease the percentage of placebo patients. So you can do what is called a 2-to-1 or even 3-to-1 randomization. So for every one patient who gets a placebo, you have 3 or 2 patients getting active drug. It seems a really attractive proposal at the time, and I know that a lot of patients and patient organizations ask for it because they prefer it. My response to that is that every time you do that, so if you go to a 2-to-1 randomization or a 3-to-1 randomization, you increase the number of patients overall in the study required to show statistical changes. Why is that important? It's important because if you're running a patient that requires 80 patients, or you're running a study that requires 120 patients in a rare disease, finding those extra 20 or 40 patients can mean the difference of 9 months maybe to find all of those patients. So you delay really the availability of the drug and creating the evidence that you need to bring this drug to the market. So it's a bit of a double-edged sword. It seems very attractive to include more patients on placebo percentage-wise, but we do pay for that in the end, and I don't mean financially. We pay for that in terms of time that it takes to get a data readout and to be able to move forward. The thing we can do as pharmaceutical companies, I think, to make this a little more attractive, or not attractive is maybe the wrong word, but a little bit more reasonable for patients, is that we really try to make it possible as soon as possible to switch to an active drug. So ideally, one would say, as soon as you've obtained sufficient evidence that the drug provides a benefit, one could stop the trial and switch all patients to active. And you can even do that at the end of a randomized period. So even while it's not read out yet, while you're waiting for the readout, patients could be switched already to active drug. And there are several study designs that allow that to be done. There are some things called adaptive designs that are quite smart now and minimize the time that patients spend on a placebo. Go to the next slide. Dan, please interrupt me or let me know if I'm being too slow. But this is, as I said, it's an important topic to me. So what can we do as an alternative? What could we use as an alternative or supportive tool to a placebo-controlled arm? We could use patient registry data. We could use retrospective and prospective natural history studies, data sharing projects, real-world data, and PROs, which are patient-reported outcomes. So this is what the patients tell us about the course of their disease, as well as what is published in the literature. All of these are potential sources to say, if you have all of these consistently telling the story of a rapid decline of the disease over 1 year, and one could then argue, well, if we then apply the drug, you don't need a placebo really, because all the drug needs to show is that it is different to what is out there in the published literature, what natural history studies have shown, or what patient registries have shown. And it's a very tempting thing, and we will not stop trying to use this with the regulatory agencies, because I personally believe that this is a very useful approach. So far, the regulatory agencies are very skeptical of this, and we haven't really had much luck when I've proposed this twice with a prospective natural history study. So really real-world data showing we followed patients for a good few years, and in comparison to this cohort, the cohort that is treated has done differently better. And this approach for phase 3 has not been allowed. If you recall, phase 2 is more for dose finding, and in there we have been able to do it. But when it comes to phase 3, where the regulators tell us what they need in order to approve the drug, we've not been able to do this. And there is a pitfall here. If the data is a bit older and there are new techniques, for instance, with respiratory therapists giving better advice on how to improve breathing, or physiotherapists come up with different exercises to strengthen or to maintain flexibility for that bit longer. This might not be reflected yet in old data, or other medication that is not considered to be a very effective treatment, but still has some effect on the course of the disease. Also, those may not be reflected in the data. So you have to be very sure that when we propose something, we have to be very sure that we are comparing really like to like very directly. If we go to the next slide, I think we so this is just a little word on clinical trials in the 21st century. As you've probably gathered by my appearance and receding hairline, I'm of the older generation. So I'm not the best with, especially with the second one, the online educational tools, but we do have now access to a lot of things that we can do in clinical trials to enhance the way they are conducted, to make life easier for patients and for patients and for family, parents or caregivers. And the first one is possibly of those the most important. We can now, with telemedicine or telecommunications, do more home assessments and data collection from home, thus preventing or reducing the need for in-person visits. And that can be very, very useful depending on the conditions. And the last one is something I've already touched on. We can do interim analyses. We can do adaptive designs that decrease time for patients on placebo. And that's something that we as a company are looking at. I've been doing that across various companies for the last few years. I think we're nearing the close. If I could just have the last slide, then yes, that's my big thank you to Dan, to everyone, to Toni and to Will, who've invited me and let me talk, and to you for your patience in listening. Thank you. Thanks, Dan. I see you back.
Yeah, thanks, Chris. That's really interesting. And actually, that's the first time personally I've seen it really clearly defined about the 1-to-1, 2-to-1, 3-to-1 ratio, and actually the impact it has going from 1-to-1, 2-to-1, 3-to-1 on time. I've never had it explained in that way, actually. So that was really interesting to understand. Obviously, you have lots of challenges and decisions you have to make, and there is so much information out there you're trying to bring together about the trial design, and it's a very challenging environment and decisions you guys have to make on the clinical side. So hopefully, the guys watching this have been able to learn a bit more about placebos from your perspective and industry, and how you've been developing these studies. So You'll join us very shortly when we have a Q&A. So I'm going to bring back Will, and Tony's going to join us. And this next kind of 10 minutes, 15 minutes or so, is just going to be a conversation to Will and Tony talking about the whole decision making about taking part in studies that have placebos, kind of Will's personal perspective, Toni from her experience in advocacy and actually helping guide people on the decision making process. It'd be good also to touch on maybe information, access to information. Have people felt they've had all the tools to make those decisions? So I'm going to drop off. I'll leave you guys to run through that for 10 minutes or so. So thank you.
Thank you, Dan. And thanks, Will, for speaking earlier and for telling us about your unique perspective as a doctor, a father, and as a patient advocate. I'm really interested to know more about this and how it might have impacted your own decision-making when it comes to placebo-controlled clinical trials.
Thanks, Toni. So obviously, for those who were listening when I was speaking earlier, I talked a little bit about my experience with my son and a clinical trial that he was involved with. In a way, that was quite a unique trial because this was a drug that was delivered by a lumbar puncture. So a lumbar puncture is an injection into the lower part of the back, which you are obviously, this is not just simply popping a pill in, but as part of that trial, they wanted to maintain a control arm. The proportion, and in fact, this was a 2-to-1 randomization. So two for every 3 patients, 2 were on drug, 1 wasn't. And the one that wasn't on drug went through the whole process as if they were going to have a lumbar puncture, but without the lumbar puncture being performed, and obviously, therefore, no drug being given. So that for my son, because he was about 7 years old during the trial, 7, 8 years old. He actually had a very brief general anaesthetic. We left him in the anaesthetic room, and this was performed every 2 weeks and went on for 18 months, where we travelled to a Birmingham Children's Hospital, and we live in Yorkshire. So it was a reasonable drive. So we would travel there every 2 weeks. He would have a brief general anaesthetic. They would then perform the lumbar puncture or not a lumbar puncture, because obviously we were blind to this. Obviously the people in the room weren't blind because they knew. And then he would then recover back on the ward afterwards and have a little plaster over the spot on his lower back where either a needle would have gone in or a needle hasn't gone into his back. The skin was simply marked. And so that was quite a unique, because this was what we would call a sham control. So rather than a true placebo, because he wasn't receiving, they didn't actually do a lumbar puncture and inject some salty water into his back or anything like that. But it was a sham procedure as a control or the equivalent of a placebo.
And could you tell us more about your decision-making process in joining that trial and the thought that must have gone into that?
Yeah, no, of course. So I guess it kind of comes down to why do people get involved in clinical trials? So so in the case of Niemann-Pick type C, but also the gangliosidosis as well, these are significant diseases which are debilitating and neurodegenerative and cause lots of problems. So we know that the course of these conditions, although quite varied from one patient to another, is progressive and significant. So we knew that, and in the case of this drug, there was quite a lot of evidence already for its likely benefit. So, I think sometimes when people enter into clinical trials, often it is sometimes perceived as being a way of accessing a drug which for whatever reason they perceive as having promise. So in this case, as I said, it has actually been used outside of clinical trials in humans before, which is obviously not the case with all drugs. And at that time, we thought that was the best chance of giving benefit for Sam. I must admit that the nature of the way that trial was performed, and then having a sham is a pretty extreme thing. This wasn't a benign thing with Sam having for us simply to give him a sugar pill every day at home, which would make very little impact to our day to day. Every 2 weeks, essentially, Sam was off school for the day of the procedure and the day after, and he went through that remarkably. It was all very straightforward. He never really had any problems with the general anaesthetic or anything like that. But it was a huge undertaking for our whole family. So, I think the individual circumstances of each family are unique, and we were very much at that point at that time where Sam still had a lot to lose, and quite a lot to gain by a timely delivery of a drug which we hoped was going to make a significant difference. So in a way, it was something that we were prepared to do. Now, that may not have been the case even for us had that same question been asked a few years later.
Yeah, I think you make a really good point about thinking about and considering the impact of participating in a clinical trial before enrolling. And I just wonder about your opinion in regard to the information that's available to families and the discussions that take place beforehand. Is there enough information, and is it the right information that families receive beforehand?
So I think the information is normally given by the physician leading the clinical trial at the centre. It's their responsibility to inform and take adequate consent, obviously with the support of organisations like NPUK and CATS and the sponsor of the trial. You can provide information. I think there's two layers. There's obviously you want to have as much information about the drug that you're receiving as you possibly can to make as informed choice as possible. But also you need to really think about, see the study protocol. So you really get an idea of how's that going to work for me? If this is saying I need to go once a month to Great Ormond Street on a Tuesday to have a day full of assessment, is that going to work with everything else and run forward? And it is something I can sustain with my work and my children's work, etc., for that year. And the answer to that is yes, and the benefit or the perceived benefits are still great, then obviously it's something that you might go along with. But all the while acknowledging, as Chris said in his last talk, these are experiments. So they're being done because we don't know that the drug works, no matter what. And we've already talked so much about why trials done this way, to minimize bias and to make it as fair a test as possible. Almost always the information you gather before, no matter how systematically collected it is, often can have biases intrinsic to it. So in the case of the drug my son was on, there were a number of children receiving this in different countries, and certainly in the US, outside of clinical trials. But I was very aware that the promising information I was being received was intrinsically biased by when you're chatting to people and looking on social media, people say the positive things. They don't say, so you're getting a bias in what you're hearing. And I was very aware that was the case.
You've already made the point, which I think is a good one, to link in with your patient support groups, such as the CATS Foundation or Niemann-Pick UK. But what other advice would you give to families who are considering taking part in a placebo-controlled clinical trial?
So obviously linking into those, I think often, certainly in the case in the UK, sometimes not every clinical centre has the clinical trials running, so it may not actually be the centre where your routine care is under. So actually having a really candid and clear conversation with your own physician, so in the case of my son, Sam is under the care of the Manchester team, but the clinical trial was in Birmingham. So it was really informative for me to be able to actually speak to the Manchester doctors, because actually there was a choice when Sam was being recruited between two clinical trials at that point, to get their views and insights into what they thought would be most beneficial, as well as and I think that personally I found that very, very helpful, as well as potentially speaking to other people who've experienced albeit not necessarily that drug, but similar clinical trials. So if this was a case of people having regular lumbar punctures, getting a real understanding of how that might work and the challenges of things like that.
Thank you, Will. Thank you so much for sharing your experience with us. I think we're moving into questions. Is that correct, Dan?
No, we are, Tony, and thank you. I'm going to bring Chris in as well to the conversation. So, Chris, you just have to unmute yourself again. What listening to you talk was I find very interesting. We personally were in the position of there were no studies when my daughter Emily was alive, so we never had any of this kind of potential opportunities to enter clinical trial, then to think about the decision-making process. But what we really found useful over the years, and Tony, you've been very instrumental in us personally, as a family, as an organisation, is that whole peer-to-peer support model where we support each other through those journey of providing care. Did you think, Will, at the time you had enough access to that peer-to-peer support of people knowing what you would have to potentially go through in terms of making that decision, or did that not exist at the time?
No, I mean, I think it had been certainly in the case of our specific disease, there had been quite a hiatus between that clinical trial and the preceding one, which had been quite a long time before, and that was an oral therapy. So, it was quite a unique form of clinical trial. So, no, I wouldn't say we did. I think the situation is quite a bit different now, in that we've now had a period of time where there's been a number of clinical trials, so people have, members of our community have gone through this, and the other thing is that certainly the LSD collaborative, which you put at the start, now across our disease area, there've been quite a number of Sam's trial that he was involved in is not only the only intrathecal trial that's run in our disease space. So, actually, in fact, by collaborating across similar diseases, you can get a breadth of experience.
That's very interesting. I think the more I think about this, I also think about the challenges that industry have. And I know that Tony, myself, and obviously you as well, Will, have been involved with companies over the years when it comes to the trial design, making those decisions. So, Chris, it'd be interesting for everyone to understand how you manage the opinions that you get from advocacy groups and patients when it comes to designing these studies, and how do you manage that process?
That's a really interesting question, and it is actually, or my response to that, I think, is largely reflected in the fact that we have, we're a small company, so we have one person, but some of the larger companies have entire departments now that are involved with patient liaison and that are dedicated to really understanding patient needs. So what we do is at meetings, either meetings that are actually run by patient organizations like yours themselves, or sometimes at disease conferences where there is a presence and there's an interest from patient organizations also. We organize meetings specifically with the purpose of understanding the patient perspective. And there we try to have as open as possible sessions that help us understand what we can do, that we can then take back to regulators, what we can do within the framework that we have to make these studies easier and more acceptable to patients. And maybe one additional point, I know that you, Dan, and you, Toni, as well, have actually been, I believe, at regulatory interaction, at regulatory bodies yourselves. So we would invite you along, for instance, or invite, in the last instance, it was at the FDA, so it was a US colleague of yours who came and helped us put the patient viewpoint forward as an invitee of the company. So those are the ways that we can understand better and therefore implement better as well.
That's interesting, because another thing that I know you have to challenge with as well is a different viewpoints of the regulators, as there may be something the FDA insists on that EMA doesn't. then another regulatory body doesn't. So how do you manage that process? Because you can't obviously design 3 completely different studies based on it.
That's also really a very pertinent question, because sometimes we have to. I have been in situations where we've had a European and a US study where it was just impossible to get agreement. Very often we can negotiate to finally agree on the same designs and endpoints, but there will always be instances where that's very difficult to achieve. One of the things that we also get feedback on from patients is that they have to do an awful lot in 2 or 3 days. Even they have to stay. Will, you were talking about the burden on the family to go every Tuesday for a day, sometimes a day and a half. The children or patients, don't have to be children, but are really knocked out afterwards, maybe miss another day of school. Is very impactful stuff. So we try to minimize what we call the investigational burden also on patients, whether they are children or adults. And that is sometimes also difficult, because if we have, for instance, the US colleagues saying, well, we really like, I'll give you an example, we like the modified SARA scale, it's done in a certain way. But the Europeans say, no, we really don't want you to modify it at all. We'd rather keep it like that, keep it as the original was intended. And then you find yourself with, well, unless you want to run 2 studies, you have to do both. And SARA is not a particularly difficult scale, but you can imagine translate that to other investigations where you end up doing things, sometimes doubling up on certain investigations, just because otherwise you are forced to run 2 studies. And that can really be quite difficult. We want to avoid that. It's also, again, a question of speed of recruitment. In rare disease, it's critical to believe that one can, in very rare diseases, run 1 European and 1 US study, and both of them will have sufficient number of patients to show a really big clinical difference. It's an illusion in the very rare disease space. So it's a real
interesting. That's interesting. I think, Toni, the question that comes to me for you is that Niemann-Pick UK, very good in your social media, your communication, how you interact with your families. Are there lessons that we could learn and share with our different communities on these sorts of subjects? So on the placebo, is there something out there missing? Do we need to be looking at, maybe we've done a webinar, but do we need to have events or bringing more groups together to discuss this topic? What do you think?
I think you're right, Dan. I think we do need to do more. And yes, we already do a lot, but we need to keep on repeating ourselves. And, the reason for that is that we have new families joining us all of the time, and we have different clinical trials with different scenarios and different products coming along. And it's really important to revisit the information at regular intervals to make sure it still stands up, but also to make sure that it's reaching the widest audience possible. So I think we can do more to communicate in different mediums, such as today with the webinar, perhaps produce resources that can be shared more widely across our websites and through the LSD Collaborative, and also through our patient conferences, so that we make sure as patient organisations that we are helping families to make informed decisions about what happens next, in particular around this topic.
I think that's a great idea, and obviously I think it's a great kind of deliverable out of this webinar, and potentially the more we're going to do throughout the next year or so about the resource we can produce to the community, because I think this is a great thing for people to be able to watch, but sometimes we need something either printed, digital, or a media where they can digest it in a lighter form in the home setting. So I think for us, me and you, Toni, that's a challenge we need to sit down and plan for. And I think this topic is very interesting, as Chris has been talking through with Azafaros. We have studies happening now with the placebo arms. We're going to be asked these questions, and I think it's very important we have the right answers. I think what we've touched on here about actually the trial design, why these decisions are making, and then actually the decision process that Will's been able to share for parents is absolutely vital. I think for us as the organisations, we need to take that on board, and we can then give that back to our community in a format that's really suitable for them. So before we close, I don't know if there's any final comments from anyone before we finish up. I know I'll hand over to maybe Will first. Got any final thoughts on today's topic?
Just that, I think it's been a really useful exercise, and I'd echo what Toni says, that important topics like this, keep revisiting them is a really useful thing, and there's no one there's no kind of right or wrong decision. Certainly when you're contemplating it yourself, and it's very personal. Just because a family, another family has decided it to go one way, it may not be suitable for you, and they can be very unique. So, gather as much information as you can. But if it's, the decision that you make will almost certainly be the right one for you.
Fantastic. What about you, Chris? Any final thoughts?
Just can only echo that it's an interesting topic that I think is not going away either. So to revisit it every now and then, to just refresh the thinking, maybe see what else is out there that we can do as an alternative to the current approach that we have. It's really important, and that we work together as well across your organizations and our industry.
Fantastic. And Toni, final thoughts from you?
First of all, I'd like to say thanks, Dan, to you and Will and Chris for a very informative and interesting discussion this afternoon. And I think my reminder to the audience is to ask questions of your patient organisation and to approach us with any questions you have, not just about placebo-controlled clinical trials, but other areas that you may wish to see information produced, and then we can help to give that information to you.
Fantastic. Well, thank you everyone for joining, for the great presentations, the great talks. We'll have this available online probably later today, and we'll share it out on our various channels. And we'll let you know when we're going to hold the next joint webinar. So thank you again, everyone. If you have any questions, please just reach out to me or Toni. You have our emails. So thank you, everyone.
Thank you.